In Chapter 5, we will be discussing the structure and function of large biological molecules, or macromolecules, as well as the smaller molecules from which they are built. All living things are made up of four classes of large biological molecules: carbohydrates, lipids, proteins, and nucleic acids. Within cells, small organic molecules, also known as monomers for single units, if you will, building blocks, are joined together to form these larger molecules. That results in the construction of macromolecules, which are large molecules composed of thousands of covalently connected atoms. As we look at the different types of macromolecules, I want you to continually remind yourself that the way in which their atoms come together via bonding to form these molecules results in very specific three-dimensional structures that facilitate very specific functions in an organism. A polymer is a long molecule that consists of many similar building blocks. These small building block molecules are called monomers, as I mentioned previously. Three of the four classes of life's organic molecules, or macromolecules, are actual polymers that are made up of many smaller units. These include carbohydrates, proteins, and nucleic acids. The large carbohydrates are called polysaccharides. And these are true polymers made up of many of the smaller unit, or the building block, made up of many monomers, hundreds or thousands of monomers that come together to build the polymer called a polysaccharide. Proteins are also polymers. The original or the initial form is that of a polypeptide. So when several hundred or so monomers called amino acids are joined together, they form a polypeptide. That will then fold into a three-dimensional form and perhaps come together with other polypeptides to form a specific protein. Nucleic acids include DNA and RNA. These are our two types of what we call polynucleotides. Again, these are polymers built from hundreds or thousands or even millions, in the case of DNA, smaller molecules called nucleotides. Thus, nucleic acids are polynucleotides, or many polynucleotides, many nucleotides that have been joined together to form the nucleic acid. So carbohydrates, proteins, and nucleic acids are three of the four groups of biological molecules, and these three groups form true polymers. They are chain-like, and they consist of many similar building blocks that, again, we referred to as monomers. The fourth class of biological molecule, that of lipids, are not true polymers, but we are still going to organize our lipid discussion into what are the smaller molecules and what are the larger molecules. For now, let's consider this relationship between monomers and polymers. A condensation reaction, or more specifically, a dehydration synthesis reaction, occurs when two monomers bond together through the loss of a water molecule. So I want you to remember this term, dehydration synthesis reaction. This is the type of chemical reaction that creates new covalent bonds between monomers, and this must happen over and over again in order to build a polymer. This does not just happen spontaneously in a cell. Enzymes facilitate all dehydration synthesis reactions. So if we consider a little molecule called a monomer, And there's a few atoms here stuck together, and we'll say there's a hydrogen also as part of that molecule. And then we're going to take another monomer, another small molecule that has an oxygen and a hydrogen attached to it somewhere, plus other atoms. In the course of a dehydration synthesis reaction, a very specific enzyme will grab these two monomer substrates and join them together by a new covalent bond. So that solid line indicates a new covalent bond has formed. In the process of creating that new covalent bond, a few atoms are lost in the form of water, H2O. So as we're building these new bonds here, we are also losing water in the process, and you can see where those water atoms came from. That's why we call this dehydration synthesis. D for removal, hydration for water, so removal of water, and synthesis is to build. So removal of water to synthesize or build a new bond. And we've taken two monomers and joined them into now a larger molecule that's called a dimer. It has two units stuck together. Polymers can also be disassembled back into their monomer units by a different type of reaction called hydrolysis, or water to break. This is essentially the reverse of the dehydration synthesis reaction shown above. Another term for hydrolysis is digestion. Bonds are broken so that large molecules are chopped up into their smaller units or monomers. As we move through our discussion of the various biological molecules, I want you to periodically remind yourself about the monomers that come together to build each polymer specifically. Recall that the monomers are the building blocks or the smaller molecules. Mono is in reference to single units. And if they are joined together by many dehydration synthesis reactions, we can get to the larger macromolecule that is called a polymer and is made of many units. Keep in mind, though, that we can also go in the opposite direction. For example, when you eat a meal. You might eat a lot of protein in your meal, and that would be over here in the polymer side of things. And then enzymes in your digestive system would break that protein down, that polymer, by hydrolysis reactions back into the smaller monomers, back into the amino acids in the case of protein. As we learn about the different monomers and polymers, keep in mind that monomers are always the small molecules, and they are common to all organisms. They are common to all organisms. However, these smaller molecules can be ordered in very specific ways or joined together by dehydration synthesis into very unique molecules, unique macromolecules or big molecules that we can call polymers. So the linear arrangement of monomers coming together will determine the structure of the polymer that is built, its function for that organism, and it will give that organism unique characteristics. So the polymers are unique for a given organism. However, they are built from very generic smaller molecules that are common to all life. This slide is simply representing dehydration synthesis reactions versus hydrolysis reactions. Dehydration synthesis is shown in the upper region, and hydrolysis is illustrated below. Starting at the very top, we can see that we've already had a few dehydration synthesis reactions because we have a molecule that is already consisting of three monomers. So there were actually two dehydration synthesis reactions previously to create these covalent bonds between the monomers. Now another monomer is going to be joined. So a very specific enzyme will grab these two substrates and by removing two hydrogens and one oxygen, in other words, by removing water, a new covalent bond will form that links this new monomer to the original. We now have a short molecule consisting of four monomers, and we're on our way to building a polymer. Polymers typically consist of hundreds or thousands, or in the case of DNA, even millions of monomers that are joined together by covalent bonds. If we wanted to take a polymer and digest it into its component monomers, we would need a different type of enzyme, and this enzyme would use water to break the covalent bond, thus producing monomers. This is a hydrolysis reaction using water to break bonds, also known as digestion. We will now begin taking a closer look at some of our specific biological molecules, starting with the topic of carbohydrates. The term carbohydrate is very general. It can refer to the monomer or the polymer of the carbohydrate. So we need to apply more specific terms such as monosaccharide or polysaccharide to know if we are discussing a carbohydrate monomer or polymer. Carbohydrates include sugars and the polymers of their sugars. The simplest carbohydrates are called monosaccharides, or single sugars or simple sugars. The example I want you to remember is glucose, and that is shown to the right. Glucose has the formula C6H12O6. There are other molecules that have these same atoms and in the same numbers, but in order to be glucose, the atoms have to be arranged in a very specific way, as shown in this picture. Typically, glucose and other monosaccharides will form ring-like structures in an aqueous environment. Since the inside and outside of cells are aqueous environments, we expect to see glucose in this ring form rather than the linear form shown above. Glucose is a very common monomer that is used in the process of building the larger carbohydrate called the polysaccharide, which is a polymer built of hundreds or thousands of smaller units, such as glucose in some cases. Monosaccharides have molecular formulas that are usually multiples of C to 2 H's to O. In other words, the carbon to hydrogen to oxygen ratio in a monosaccharide is typically 1 to 2 to 1. One carbon for every two hydrogens for every one oxygen. And as you look at the formula for glucose, you will see that it does meet that criteria of 1 to 2 to 1 ratio between these atoms. Glucose is the most common monosaccharide or monomer in the carbohydrate world. And as I mentioned previously, though glucose is often drawn as a linear skeleton in an aqueous or water solution, such as inside a cell, glucose and other monosaccharides typically form ring structures. Monosaccharides in general serve as fuel for cells, so energy, to power all of life's work, and they also serve as raw material for building larger molecules, for building polysaccharides. And of course, this would require dehydration synthesis reactions. We can classify monosaccharides by the number and location of carbons in the monosaccharide, as well as the location of the carbonyl groups. And we'll take a look at that on the next figure. On this figure, we have three names listed on the top: trioses, pentoses, and hexoses. These names are in reference to the number of carbons that make up each monosaccharide. So trioses have the prefix tri, so there must be 3 carbons that are found in each triose. Pentoses, that's in reference to 5, so there must be 5 carbons in every pentose monosaccharide. Hexoses have 6 carbons per monosaccharide, and so forth and so on. The ose suffix in each case is in reference to sugar. Along the left-hand side, we have a couple other names, aldoses and ketoses. And this is in reference to the carbonyl group, whether it is on the end of the molecule attached to a hydrogen atom, or whether it's embedded in the molecule attached to 2 different carbon atoms. So these are just some ways that we can classify and categorize our different types of monosaccharides, or the monomers in the carbohydrate category. Glucose is our point of reference. Glucose is our example monosaccharide that we will study further. Glucose is a hexose because it has 6 carbons. As mentioned, we will focus on glucose as our example of a monosaccharide because glucose is so important in so many larger carbohydrates named polysaccharides. In this illustration, we can see linear forms of glucose and then the process by which it forms a ring-shaped structure in an aqueous or water-based solution. Here is the abbreviated ring-shaped structure of glucose. Note that it has the molecular formula C6H12O6, but in this abbreviated structure, you don't see the 6 carbons. That's because every point in this structure represents a carbon, even though it is not shown. Get used to seeing those points in biological molecules or organic molecules and remembering that wherever you see a point, there is an inferred carbon atom. Here, once again, we are looking at dehydration synthesis reactions, which are required in order to build a disaccharide, a trisaccharide, and eventually a polysaccharide. So these are removal of water to build new covalent bonds to build our macromolecules. Building a polysaccharide has to start somewhere. For example, we need to have 2 monosaccharides that are first joined together by a covalent bond. And we're looking at an example of that shown above. Here we can see one monosaccharide called glucose is going to be joined to another glucose monosaccharide. So in this chemical reaction, we have our 2 substrates, which are both glucose. The atoms highlighted in blue are going to be removed during the course of the chemical reaction by an enzyme, a very specific enzyme that only does this chemical reaction. That enzyme will capture the 2 glucose monomers, remove 2 hydrogens and an oxygen from them, as shown over here, and then create a new covalent bond linking those 2 molecules together. So here is that new covalent bond. The resulting product is what we call a disaccharide because there are 2 sugars making it up. A disaccharide in this case called maltose. Maltose is in germinating grain, and it's really important if you want to make beer. So you'd mix your water and your hops and your barley together with yeast, and then the yeast would ferment the maltose into ethanol. Another dehydration synthesis reaction is shown below. This time the substrates are 1 glucose and a different monosaccharide called fructose. Fructose also has the molecular formula C6H12O6, but you can see the atoms are arranged a bit differently in this monosaccharide. In the course of this dehydration synthesis reaction, a different enzyme will grab glucose and fructose, remove a few atoms from them, and create a new covalent bond, linking them into a different disaccharide called sucrose, or table sugar. The type of reaction illustrated on the previous slide must happen hundreds of times in order to build a large carbohydrate that we refer to as a polysaccharide. Polysaccharides are the polymers of the simple sugars, and polysaccharides have very important storage and structural roles for a cell. Storage in reference to energy storage, and structural in reference to protective functions. Some examples of important polysaccharides are shown on the slide. First of all, energy storage polysaccharides that you need to know. Starch is one example. Starch is a polysaccharide made by plants, and starch is built entirely of glucose monomers that have been joined together by dehydration synthesis reactions. Glycogen is another polysaccharide made by animals, such as us. Glycogen, like starch, consists entirely of glucose monomers joined together by dehydration synthesis. However, the glucose is organized in a different fashion so that glycogen has a very different three-dimensional structural shape as compared to starch. It's a very different molecule, even though the monomers are the same. Humans and other vertebrates store glycogen mainly in our liver and muscle cells, energy reserves. A third example of a polysaccharide that has a structural role for an organism is that of cellulose, and cellulose is made by plants. Once again, cellulose is an example of a polysaccharide that is made entirely of glucose monomers, and once again, they are joined together by dehydration synthesis, although in a different fashion than the previous examples above, to create a unique type of polysaccharide. Cellulose is the major component of the tough cell walls of plants. So all cells, as you know, have an outer layer called the cell membrane. Some cells, such as plant cells, have an additional structure outside of that cell membrane that's very rigid, and we call that the cell wall. And the major component of plant cell walls is cellulose. On this slide, we are revisiting the three examples of polysaccharides that were presented to you on the previous slide. Notice the photograph on the top left. We see a person that has dug up some fresh potatoes from the garden. If we are able to look at a little region of this potato and zoom in on it with a microscope, of course, we can finally see cells that make up that potato tuber. Inside those cells, we will see a special type of plastid that is called a starch granule. Inside that starch granule, we find lots and lots of the polysaccharide starch. And this is the way the plant is storing energy. So if we look at a starch granule inside a potato cell and zoom in on it, we can see this branching molecule, all of this shown in green. This is starch. And as you zoom in on it closer and closer, you can see that it's built from many hundreds or thousands of glucose monomers that have been linked by dehydration synthesis to form this large macromolecule, a polysaccharide known as starch. If we look on this person here and zoom in on a muscle cell of that person, of course using a microscope, we have to have a microscope to see cells, we could see that within this muscle cell we have glycogen. And glycogen is the polysaccharide that we use to store energy. So glycogen, although built from glucose monomers as well, has a very different structure to it compared to starch because of the way the glucose is organized. However, both glycogen and starch are used for energy storage in different organisms. And then finally, if we look at this leaf in the photograph, here is a potato cell leaf, so the photosynthetic part of the potato plant. We zoom in on that plant leaf with a microscope and look at those cells. We can now see the chloroplasts inside the cells, and then a cell membrane, and outside of that, a cell wall. And that cell wall is what we're really going to zoom in on further. This time it will require an electron microscope. This picture here was taken with a scanning electron microscope so that we can see the three-dimensional structure of that cell wall material. And in that cell wall material, you're going to find cellulose. Cellulose is a polysaccharide, and it is the major component of plant cell walls. If we zoom in on one of these little fibers here of the plant cell wall material, this is called a microfibril, and this is just cellulose molecules that are bound to other cellulose molecules with hydrogen bonds. And so to see that structure, we'll zoom in even more. And we can see here that here's a long branching cellulose molecule, another long branching cellulose molecule, and a third down below. And these dotted lines that are linking them together, these are hydrogen bonds. You learned about those previously. Notice that cellulose has a different structure compared to both glycogen and starch, even though it is made up of glucose monomers as well. It also has a different function for the cell. This time we're looking at a structural or protective function rather than energy storage. Like starch, cellulose is a polymer of glucose. That means it is a big molecule made up of many, many repeating units of glucose. So perhaps thousands of glucose monosaccharides joined together by covalent bonds that we call glycosidic linkages. Recall that all of these covalent bonds are a result of dehydration synthesis reactions to build the cellulose. Starch and cellulose are both made of glucose, but the difference is how all those glucose units are joined together, and actually the forms of glucose that are used to build these two polysaccharides. Starch is built from a form of glucose that we call alpha glucose, whereas cellulose is built from a form of glucose that we call beta glucose. Both versions of glucose are C6H12O6. The difference is in the location of this hydroxyl group, or this OH group. That's the difference, is where it is positioned. And that affects the final structure of our polysaccharides when the beta glucose is linked together versus when the alpha glucose is linked together. Very different structures. Because those glycosidic linkages differ, enzymes that digest starch, enzymes in our digestive systems that break down the potatoes we eat, cannot break the bonds in cellulose. So our enzymes can digest starch or perform hydrolysis, chop it up, but our enzymes cannot break down cellulose. So when you eat plant material, the enzymes in your digestive system cannot hydrolyze those bonds in the plant material cell wall. And therefore, cellulose in our food passes through our digestive systems intact. And we call that insoluble fiber, or dietary fiber, if you will. Dietary fiber is really important for keeping our food moving through our digestive systems. There are some organisms that have the right enzymes for digesting cellulose. Some microbes in particular have the correct enzymes necessary to break down cellulose or to perform hydrolysis, if you will. Water to break bonds, digestion. Many herbivores have these microbes. Cows, for example, have microbes in their digestive system that help them break down the plant material they eat. The microbes inside the cow have a very important mutualistic relationship with the cow. The cow eats the plant material, and the microbes hydrolyze the cellulose so that the cows can obtain energy and building blocks from the cellulose. Termites also have microbes that have a symbiotic relationship with them. Do you remember the type of microbe we looked at in the lab? It was a protozoan called Trichonympha. And Trichonympha helps termites break down the wood material, the plant material that they eat. Another symbiotic relationship called mutualism. We have microbes in our digestive systems that help us with various processes as well. However, our microbes do not digest cellulose for us. Another example of a structural polysaccharide is chitin. Notice how I pronounced chitin? It is not chitin. Chitin is found in the exoskeleton of arthropods, such as insects, spiders, crustaceans, those organisms that have that hard outer shell, if you will. Down below is an arthropod that is shedding its exoskeleton so that it can grow. This is a cicada shedding its exoskeleton. Chitin also provides structural support for the cell walls of many fungi. So besides arthropods, fungi have this polysaccharide as well. Fungal cell walls, of course, exist outside the cell membrane. So a fungal cell will have a cell membrane, and then outside of that, it will have a rigid cell wall, similar to plants with their rigid cell wall. But whereas plants use cellulose in their cell walls, fungi use this different polysaccharide called chitin. So chitin is a structural or protective polysaccharide that is used by different organisms. Chitin is also used to make a strong and flexible surgical thread, as shown in the photograph to the right. The monomers that are assembled into chitin are shown here on the left. Here's one of the monomers. This is N-acetylglucosamine, a little different from the glucose monomer we looked at previously.